Good morning, class. Today we will study genetic engineering, that is recombinant DNA technology. In short, we call it RDT. Recombinant DNA technology. Now what does this topic tell you? It tells you that we create a recombinant DNA. How is the recombinant DNA created? Recombinant DNA is created with the help of using enzymes, with the help of using DNA. DNA from two different sources are used. They are combined. That is why this technique is known as recombinant DNA technology. So what do we do here? We have direct manipulation of an organism's genome using biotechnology. What is a genome? Genome is the total DNA complement of a given organism. The total DNA that is present in a given organism. That is known as a genome. So what are genetically modified organisms? When any organism which occurs in nature is taken and modified genetically, that is, its gene or the genome is modified in the laboratory for some beneficial purposes, then it is known as genetically modified organism. An organism that is generated through genetic engineering is a genetically modified organism. In short, we call it GMO. Now what are the applications of recombinant DNA technology? Bacteria, they were engineered to produce human insulin. How do we produce insulin in bacteria? We take DNA from the human source. We take the chromosome from the human being. From that, we cut out or cleave out the DNA, that is the insulin gene. That gene is then ligated with the vector. Vectors can be many. We'll discuss it later. It is ligated with the vector. This becomes recombinant DNA molecule. Now this recombinant DNA molecule is then inserted into a host cell. What is a host cell? Host cell is any cell in which the recombinant DNA molecule can be propagated. Now the host cell, these can be of two types, cloning and expression. Again, the vectors can also be of two types, cloning and expression. What is the difference between cloning and expression, vectors and hosts? Suppose there is a DNA molecule which we want to amplify, that is, increase in number. Suppose I have 5 copies of a particular DNA or a particular gene and I need approximately 500 copies of that gene. So I need to increase the number of that gene. How do I increase the number of that gene? By cloning. This is cloning. But for an example, if we have a diabetic patient, the diabetic patient needs regular shots of insulin to control the blood sugar. Where do we get the insulin from? We can get it by cloning. How? We clone the insulin gene in a bacteria which has transcription and translation expression sequences. In that, what happens? When we put the gene in that bacteria which has transcription translation sequences, the gene which we have cloned in that bacteria can be expressed to form proteins. The protein in case of insulin is insulin hormone. This insulin hormone can then be easily purified, processed and used as medicine. The purification process here would be known as downstream processing. So we have two types of vectors, we have two types of host, cloning and expression. Now E cells were used to produce hepatitis B vaccine. I hope you know about prokaryotic cell and eukaryotic cell. The proteins which we have to clone from the eukaryote, we try to use a eukaryotic vector as well as a eukaryotic host, because there are certain problems when eukaryotic protein is cloned in a prokaryotic host or a prokaryotic vector. So preferably we use eukaryotic vector and eukaryotic host. So what is the difference? Why do we need to use a eukaryotic host and eukaryotic vector as compared to prokaryotic? See, prokaryotes, the chromosome of prokaryotes, it doesn't have introns and exons. So splicing process does not occur in prokaryotes, whereas it occurs in eukaryotes. So if we are cloning a eukaryotic gene, it needs to undergo splicing while expressing the protein. And that splicing does not occur in prokaryotes. So we need to clone the particular gene in eukaryote. Secondly, the post-translational modifications. The eukaryotic proteins need to undergo post-translational modifications for proper folding, which is not available in prokaryotes. That is why for cloning of eukaryotic proteins, we need to use eukaryotic vectors and eukaryotic host. And yeast is one of the most commonly used eukaryotic host. Plants such as cotton were made insect resistant, like Bt cotton. Bt here stands for Bacillus thuringiensis, from which the gene for resistance was isolated. Large amount of DNA can be generated by using gene cloning. So, gene cloning, gene expression is a mechanism for generating proteins, for generating large amounts of DNA by cloning and expressing in various vectors and host cells. The vectors and the host cells, they can be prokaryotic or they can be eukaryotic. What are the advantages of recombinant DNA technology? A large amount of recombinant DNA can be generated, that is gene cloning as we just discussed, and a large amount of protein can be expressed by the insert. As we just discussed, as in case of diabetic patients, we need the insulin hormone and not the insulin gene to be injected into the patient. So the hormone has to be expressed. And this is what we do in recombinant DNA technology. The first recombinant DNA molecules were generated in 1973 by Paul Berg, Herbert Boyer, Annie Chang and Stanley Cohen. What are palindromic sequences? Any idea? The sequence in both the DNA strands at this site read the same in 5' to 3' direction. Now what is the need for palindromic sequences? Why are we discussing palindromic sequences here? Palindromic sequences, these are 4 to 8 base pair sequences which are present in the DNA, in the chromosome, and these sequences can be cleaved or cut by the restriction enzymes to generate either blunt end or sticky ends. Now what are blunt ends and what are sticky ends? Blunt ends are created when the enzyme cleaves the recognized sequence from the center. So there are no overhangs generated. But when there is asymmetric cut by the enzyme, then overhangs are generated, which leads to creation of sticky ends. In case of genetic engineering, we prefer sticky ends because they are easy to ligate to each other, as well as easy to ligate the DNA insert to the vector. What is DNA ligase? This is an enzyme which covalently links two fragments of DNA by forming phosphodiester bonds between adjacent nucleotides. The reaction requires one of the fragments to have a 5' phosphate residue and the other a 3' hydroxyl group. For example, DNA ligase isolated from bacteriophage T4. Now what is the function of ligase? Why are we mentioning this enzyme here? Ligase is an enzyme. Just before this we talked about restriction enzyme, palindromic sequences. I told you the restriction enzymes can cut the DNA in two forms. Either it can be blunt or it can be sticky. When we cut with the help of restriction enzyme, now we need to join two sequences together. Which two sequences? The DNA which we have isolated from the chromosome. For example, the gene for insulin and the vector. The vector is the vehicle for carrying the DNA inside the host cell as well as replicating it. So we need to join them together. The enzyme ligase does this function of joining or ligating two fragments of DNA together. Sometimes the vector, when it is cleaved with a restriction enzyme, it might religate without taking the DNA insert. It might self-ligate, religate. So making the effort of cleaving the vector with a restriction enzyme futile. The effort of cleaving goes waste since DNA does not enter. So how do we prevent self-ligation or religation of the vector? We use alkaline phosphatase. Alkaline phosphatase again is an enzyme. This enzyme removes phosphate from the 5' end of the vector molecule, the cleaved site of 5' end of vector. So now the vector is left with only 3' OH group, removing the 5' phosphate. Thus, no phosphodiester bonds would be created, no joining by ligase will take place. So vector is now not able to self-ligate. It needs a molecule from outside, a DNA from outside to come and provide 5' phosphate. So when the DNA insert comes in and joins, only then the vector can ligate to the DNA insert and not self-ligate. So alkaline phosphatase molecules are used to remove 5' phosphate from the vector to prevent self-ligation. If vector molecules are treated with alkaline phosphatase to remove their phosphate groups, then these cannot ligate within themselves and are forced to ligate with other fragments containing 5' phosphate groups. This prevents self-ligation. Alkaline phosphatase is purified from bacteria or calf intestines. Now, we were talking about vectors. There are many varieties of vectors available. Vectors are mainly, as I told you, used as vehicles for carrying the DNA insert inside the host cell. Also, provide conditions for the DNA insert to replicate. So, we have here two types of vectors, that is plasmid based. Here we have PUC and PBr322 plasmid. The small p stands for plasmid. UC stands for University of California. In case of PBr, BR stands for Bolivar and Rodriguez. So we have two types of plasmid vectors here. Now what is the difference between these two types? PBr322 was named after the researchers who constructed it. The P stands for plasmid, BR for Bolivar and Rodriguez, whereas P in PUC19 stands for plasmid and UC represents University of California. PBr322 has 4,361 base pairs, whereas PUC19 has 2,686 base pairs. PBr322 contains ampicillin resistance gene and tetracycline resistance gene as marker genes, whereas PUC19 has ampicillin resistance gene and LAC-Z gene as marker genes. Now what are marker genes? Why do we need to use them in our vectors? Marker genes are, suppose we have a vector which self-ligates. We have a vector which has DNA insert. So we have two types of vectors available in the medium. One is non-recombinant vector, one is recombinant. Plus, when we insert these vectors inside the host cells, again, some of the bacterial host cell or any other type of host cell that we use might not take up any vector. Some host cells might take up the vector which is non-recombinant, but some vectors might take up the vector, some hosts might take up the vectors which are recombinant. So we have approximately three varieties here. The bacteria or the host cell which is not having any vector, the host cell having non-recombinant vector, and the host cell having recombinant vector. Our purpose is to select out the host cell having the recombinant vector. How do we do that? So to select out the host cell having the recombinant vector, we use selectable markers. The selectable markers which are used are ampicillin resistance, tetracycline resistance. These two are antibiotic resistance genes which are incorporated inside the vector to give it special properties. What special properties? Of having resistance against ampicillin antibiotic and tetracycline antibiotic. On the other hand, the POC19 has one ampicillin resistance gene and one LAC-Z gene as marker. The ampicillin resistance gene again confers resistance to ampicillin antibiotic. What does LAC-Z do? LAC-Z is a gene which produces an enzyme beta-galactosidase. Now what does this enzyme do? This enzyme digests or converts X-gal substrate, which is white in color, to a product which is blue in color. So the bacterial colonies or the host colonies which look blue in color, which appear blue in color, they have the vector containing LAC-Z gene, and the colonies which appear white in color, they do not have the LAC-Z gene. This helps us to distinguish between the host cell which contain the vector and which do not contain the vector. PBR322 does not contain multiple cloning site. It has unique restriction sites for more than 40 restriction enzymes, whereas PUC19 contains multiple cloning site present in LAC-Z gene. What are multiple cloning sites? We need unique restriction site for every enzyme so that we can cleave the vector and the DNA insert using the same restriction enzyme and then ligate it. It is a necessity that we cleave both the vector and the desired DNA with the help of same restriction enzyme. Why is it so? Because same restriction enzyme generates overhangs or sticky ends which are complementary. So joining together of complementary strands become very easy. That is why we need to cleave both the vector as well as the desired DNA insert using the same restriction enzyme. Now, in case of PBR322, we do not have MCS. MCS is multiple cloning site, a fragment containing palindromic sequences or recognition sequences, unique recognition sequences for multiple restriction enzymes. That is one site for many enzymes. That is one restriction site for HIN3, one for BAMH1, one for SMA1, one for HAE3, etc. So we have one site for many restriction enzymes. That fragment is known as multiple cloning site, and this is incorporated in PUC19 but not in PBR322. PBR322 contains 40 sites, 40 restriction enzyme sites separately. What are shuttle vectors? There are sometimes requirements of cloning and expressing a particular gene in prokaryotes as well as eukaryotes. But as we discussed earlier, we have vectors and hosts which can lead to progression, amplification or expression of proteins only in prokaryotes or in eukaryotes. Shuttle vectors solve the purpose of producing proteins of prokaryotic and eukaryotic origin in the same host. So the vectors which can exist both in eukaryotic and E. coli cells, these contain two types of origin of replication, selectable marker genes, and one set functions in eukaryotic cells, another in E. coli. So these are shuttle vectors. Students, We'll discuss more about this chapter in the next class. Thank you.